This Week in Microbiology is brought to you by the American Society for Microbiology at microbeworld.org/TWiM. From the American Society for Microbiology and Microbe TV, this is TWiM, This Week in Microbiology, episode 143, recorded on January 5th, 2017. This episode is brought to you by CuriosityStream, a subscription streaming service that offers over 1,500 documentaries and nonfiction series from the world's best filmmakers. Get unlimited access starting at just $2.99 a month, and for our audience, the first 2 months are completely free if you sign up at curiositystream.com/microbe and use the promo code microbe. Hi, everybody. I'm Vincent Racaniello, and you're listening to TWiM, the podcast that explores unseen life on Earth. Joining me today from Ann Arbor, Michigan, Michele Swanson.
Hello. Happy New Year.
You too. Wow, we're already 5 days into it. And it's freezing here. It must be freezing there too, right?
Yeah, it's in the teens.
Do you have a lot of snow?
Well, no, because the day after Christmas, December 26th, it got up into the 50s.
Wow.
So whatever snow we had is gone, and we just have a little dusting right now.
Also joining us from Charleston, South Carolina, Michael Schmidt.
Hello, everyone. Happy New Year.
You too. You got warm weather down there, Chamber of Commerce?
We did until this weekend. This weekend, they're using the four-letter word that begins with an S and ends in a W.
Show.
Yeah.
Yes.
There you go.
Yes.
Showtime.
We're all terrified because South Carolinians don't do well with snow.
One of these days I have to get to Charleston. Never been there.
Ooh, great
We're going to
food.
have to bring you down.
Went through the airport a few times.
You did?
No,
It's
I guess-
normally a dead end.
No, I'm sorry. I'm thinking of Charlotte.
Oh, everybody's gone through Charlotte. I think when everyone dies, they go through Charlotte or Atlanta.
Oh, boy. That's an interesting one. Well, Elio's at a meeting today, so he's not joining us. So we will forward on. And I want to tell everyone who's listening, over on TWiV, we do book giveaways. And I'm going to start that here on TWiM. I have
Cool.
a bunch of, I get books from ASM Press all the time, and I don't need them all. So I asked them, and they said it would be great if I gave them away. So stay tuned at the end of the show. I'm not going to tell you where. You have to listen. I'm going to tell you how you can win a copy of a very cool ASM Press book. Stay tuned.
I'm on the edge of my seat, Vincent.
Right. All right. We have two papers for you today. We have a snippet and a paper. And although, the lines blur sometimes, right? Sometimes they're reversed. Sometimes they're the same. But we try and make a snippet shorter than usual. And Michael is going to give us a snippet today.
Well, in honor of the Consumer Electronics Show that's kicking off in Las Vegas this week, this is the time when electronics are big. I thought I'd bring you this paper today, which is really a pretty neat topic. The paper is from one of the Nature partner journals entitled Biofilms and Microbiomes. It's an open access article, so you'll be able to see what we're talking about, and it's entitled The Eradication of Pseudomonas aeruginosa biofilms and persister cells using an electrochemical scaffold and enhanced antibiotic susceptibility. And this paper is from a group from the Jean and Linda Voland School of Chemical Engineering and Bioengineering at Washington State University and the Paul G. Allen School for Global Animal Health, also at Washington State University in Pullman, Washington. And the authors were Sajela Sultana, Douglas Call, and Halak Benal. And it's a really, really cool paper.
Michael, what is this this journal? It's a Nature partner journal? What is that?
That's what it's entitled. It has all the typeface of Nature. It looks like this was it, and it's got the moniker www.nature.com/NPJ Biofilms.
Biofilms.
So
at the bottom it says published in partnership with Nanyang Technological University.
That's right. So they are partners, and you can read more about these partnerships, but their Nature Journals is looking to partner with other institutions to support these journals. Evidently, Nature now has at least 70 different journals.
This is something.
So this university has exclusive rights to this, or they support it? Is that how it works?
I don't know. I suspect they provide some funding or infrastructure.
I see.
Yeah, but it's got all the same editorial rigor that a Nature paper does. It's a first-rate manuscript from these authors.
And it's open access, I see.
It's open access, and so you'll be able to get access to all the figures. And I was intrigued because chronic and recalcitrant biofilm infections are often chronic and recalcitrant, often requiring extremely large concentrations of antibiotics in order to clear them from a patient. To give you an example, it often takes 500 times the minimum inhibitory concentration of an antibiotic to clear a biofilm-based infections. 500-fold concentrations of antibiotics, as everyone can well imagine, begins to border on toxicity. And in fact, that is indeed what happens. It's very challenging, and you have to take this antibiotic regimen to clear a biofilm infection. And this property of chronic and recalcitrant biofilms being refractory to antibiotics is thought to result from the presence of a subpopulation of cells that are normally resident in either mixed biofilms, in which there are many different types of microorganisms, or even pure culture biofilms. And this subpopulation is referred to as persister cells that are normal residents of the community, which are nothing more than dormant variants of regular cells that form stochastically in microbial populations all the time, and they're just simply sitting there dormant. They're not spores, so they're not heat stable, and they're not resistant to all of the stressors that spores normally are refractory to, but they are indeed very resistant to high concentrations of antimicrobials. And this goes back to how antibiotics actually work, in that they're targeting essential processes to actively dividing cells. And so the antibiotic you wouldn't expect would have any effect on the cell if it's not actively divided.
And could I just add
Michele,
about persister cells? It's a way that a population of bacterial cells hedges their bets, so that if conditions are suddenly stressful or not favorable, it might kill off the replicating cells, but the persisters can live and carry on the genomic line for another day.
It's sort of like the seed bank in all those sci-fi movies where the planet is wiped out and everybody's sent to live in a cave. And it's the microbial way of preserving the species for the next generation. And generally, the persister cells represent a fraction of the biofilm. Typically, what the literature reports, it's about 0.3% of the total population. And you're going to say 0.3%, that doesn't seem like a lot. But if you consider that the average biofilm has a density of about 100 million cells per cubic centimeter, and 0.3% of that is 300,000. So you can see the genetic richness that remains. Even if all the living cells are killed off, they still got 300,000 individuals to kickstart the population again. And so now you can understand the problem of treating an infection that's in a biofilm. And these persister cells have this ability to tolerate antibiotics because they're just sitting there. And you give them the antibiotic, you kill off the biofilm, and then the antibiotic is taken away, and the persister cells come out of dormancy, and voila, things go back to the way they were. So you can immediately imagine this is not good. So this paper takes us into new territory of how to treat infections in a biofilm. And the way they accomplish this task is through the generation of the in situ generation of a potent biocide. And the potent biocide that they're generating is hydrogen peroxide. And what the paper does is it teaches us how they establish that it is indeed hydrogen peroxide that's doing the dirty deed, and these other reactive oxygen species that are a consequence of the electronic scaffold being able to generate this very potent biocide that's generated from just a very small amount of current. And so it's a really neat paper. But before we get into it, I just want to give you a little bit of a background on biofilms in case you're not familiar with them. I learned a lot of what I know about biofilms sitting next to Gordon McFeeters from Montana State University. And he and I were on an ASM committee once upon a time when we were both on the ASM education board, where we were developing the microbe library. That's one of the great ASM resources. And Gordon began to introduce me to the wonderful world of biofilms. Gordon at the time was at the NSF Center for Biofilm Engineering at Montana State, and he and his colleague, the late Bill Costerton, really were the pioneers of doing a lot of fascinating work on biofilms. And I learned that they're really nothing more than bacteria that have decided to sit down and take a rest, if you will. And biofilms form around this extracellular polymeric substances that are secreted by members of either this pure or mixed community of bacteria. And they come together as a consequence of the expression of this extra polymeric substances that form the biofilm matrix. And this is effectively how they resist the antibiotics. This EPS serves as a diffusion barrier, and in turn, it protects the bacterial communities by first limiting antibiotic penetration, and secondly, by immobilization of the antibiotic, often in this EPS material. It's effectively a giant sponge, if you will, to effectively soak up the antibiotic.
But within that sponge, that adherent sponge, there's fascinating biology. There's amazing division of labor within the cells in that biofilm. So it's more like a tissue than it is a pure culture of bacteria. So different bacteria within the biofilm are doing different jobs, scavenging nutrients or being persisters or providing structural support.
Eating somebody else's waste.
Right. So I think we're still learning about the communication and the division of labor within biofilms, but it's really a fascinating area of biology.
And, the whole genetics of persister cells, and I dug up a review that's also open access that I'm going to put into the show notes that was authored by Kim Lewis of Northeastern University in Boston in 2010. And he begins to describe how persister cells, some of the genetics of persister cells, and we haven't really talked about persister cells on TWiM, so I thought I do you all the service of putting out or at least acquainting you with Kim Lewis's review. And he wrote a really interesting review, and it really highlights the literature, albeit it was authored in 2010, so much has happened. But I was looking for something that you could, if you're interested in persister cells, you could take a look at. So the story is that peroxide enters the cell through the normal suspects. They enter through porins, and peroxide by itself can also dissolve the bacterial membrane. And as the peroxide goes in, it interacts with iron 2 and then is transformed by Fenton chemistry, generating these hydroxy radicals that are then highly reactive against thiol-containing proteins, and it effectively renders them inactive. It also, as you can well imagine, these hydroxy and singlet oxygen species that are being generated by the Fenton chemistry then go off and damage the DNA that literally breaks the DNA. It can interact with membrane proteins, and it uncouples the membrane potential. So that's how these authors lay out how they think the mechanism of their electronic scaffolding is actually taking the persister cells out of the community. And what the paper does for us is it teaches us how they were able to address this question. So I'll get into the 3 sets of experiments that they did, and the figures are fairly straightforward and easy to understand. And the first section of the results is they introduce us to this electrochemical scaffold that enhances the tobramycin susceptibility in biofilm cells. And the first section of the results helps us appreciate that this electrochemical scaffold enhances tobramycin, which is an aminoglycoside antibiotic that is routinely used to treat gram-negative infections, especially noteworthy for its ability to target Pseudomonas. And this antibiotic is an aminoglycoside, and it binds to both the 30 and 50S ribosome, preventing formation of the 70S complex, preventing, obviously, the translation of mRNA into protein. It's not active against gram-positive bacteria, with the exception of Staph aureus, which I always found a little bit peculiar. So their first experiment teaches us how they were able to introduce electrical current to their experiments, and their figures are spot on. They're easy to figure out, and their cartoons of their experimental setup are really elegant in how they were able to do this. But since this isn't television and it's radio, I'm going to try to describe it. So they first establish a fresh biofilm. This is not hard to do. Pseudomonas loves to do this. You just grow up Pseudomonas in luria broth and a sterile glass-bottom Petri plate. The biofilm forms on the glass, and the biofilm will form in about 24 hours. These one-day-old biofilms are then treated with their E-scaffolds for 24 hours, where they place a carbon-based conductive fabric on top of the biofilm, to which a titanium wire is attached. And upon applying a low current, the peroxide forms simply from atmospheric oxygen that's diffused into the liquid medium. And it then results in the electrochemical conversion of oxygen, which then generates an in situ constant generation of a small amount of peroxide that ultimately results in the elimination of this persister cell population, because peroxide doesn't need an actively growing cell in order to kill it. It just effectively is reacting with the proteins in the membrane and the proteins inside the cell and the DNA.
And the DNA and
all of the above. So their experimental setup is illustrated in the figure. And since it's open access, I also put that in the show notes. But the way I visualized it is I considered the biofilm as the ham in a ham sandwich, and the cheese in a ham and cheese sandwich. And the cheese is this fabric to which they connected it to a potentiostat, which is nothing more than a power supply that we all experienced if you played with electric trains over the Christmas holidays, or if you were an organic chemist and you played with a variac. We all remember the variac we use for our refluxing. And so they apply the current and you generate this oxygen. But the really cool aspect of their experimental design is how they were able to fish out persister cells. And the way they did that is they based it on the observation that antibiotics will kill actively growing cells. So in the case of getting persister cells, what they would do is they would grow them in the presence of, and here they used another antibiotic, ciprofloxacin, and they would grow them in the presence of Cipro for about 3.5 hours, and then they would harvest them, and you would have a population of persister cells that they were then able to do their subsequent experiments. So in their first experiment, they take us through, and it's a really simple design in which they have increasing concentrations of the antibiotic tobramycin. So they have 5, 10, 20, and 40 micrograms per mil of tobramycin, and they're just doing the viable count, asking the question how many bacteria are indeed present. So you have your fresh biofilm, and then you add your tobramycin, and you ask the question, how many cells do you kill? And I'm just going to round the numbers up to the nearest whole number because they're expressing them in logs. And so the no treatment starts at about 10 to the 8th cells per centimeter squared. So you're starting out with about 10 to the 8th. And if you add tobramycin and you haven't done any of the treatments on them other than the tobramycin, you see that the concentration drops about 4 logs down to about 10,000 cells or 10 to the 4. Then what they did is they have untreated biofilms. So these are biofilms that have been growing, and they add the tobramycin. And what you see is that the population is a little bit more refractory as time goes on, and you're only able to drop the concentration down to about 10 to the 5. So you effectively see that this persister cell enables them to go on. And when you go across the spectrum of the concentrations of tobramycin, they're effectively not very different in the untreated biofilm. When they conduct the experiment solely on persister cells, you can immediately see that the persister cell is a few are maybe killed by the initial exposure of tobramycin, but it nowhere near drops the concentration down. So the majority of that population of persister cells that they have, which is well over 10 million per CFU, 10 million CFUs per centimeter squared, it's, at that same concentration all the way out to 40 mics of tobramycin per mil. But then when they add their electronic scaffold, apply a little current and add the tobramycin, and increasing, you immediately begin to see that there's a dose response.
Michael, how do they distinguish persisters from other cells?
They isolate the persister cell from the presence of chloramphenicol.
I think it's just by definition. Whatever survived the chloramphenicol, they called that persister.
Okay. The Cipro. I shouldn't have said chloramphenicol. Yep. So,
but Michael, in that figure, what do we look at the no treatment to learn how many cells were killed by the E-scaffold alone?
No, that's not present. That's in one of their previous manuscripts.
Okay, so we don't really know how much, whether this is a synergistic killing or it's an additive killing.
No, we don't know that. That's in their previous work. And since this was only a snippet, I didn't want to introduce that variable into the mix. But I think you'll believe that the E-scaffold is actually taking out the persister cells. And that's the second argument of their paper or their results sections, is that the electrochemical scaffold then eradicates the persister cell. And the way they did that particular experiment is that they isolate the persister cells. And again, they teach us how to isolate persister cells, exploiting the operational definition that persister cells are refractory to antibiotics, and they use Cipro to do it. So here the selective pressure is to grow the biofilm in the presence of Cipro for 3.5 hours. They then disrupt the biofilm, harvest the cells, and operationally, though, that fraction is the persister cell population. And what they determined is about 50,000 of the 20 million cells that they harvested, comparing it to the control, were indeed persister cells, which is what the literature reports for persister cells being what fraction of a population. And again, the way the experiment is set up, they have the control in which nothing has been done. And so you have total biofilm cells that have then undergone 6 hours of treatment, and then the persister cells. And for all intents and purposes, there's no difference in that initial load between total biofilm and the persister cells. And that's where you determine that the persister cell population is about 0.31%, or 50,000 of the 20 million cells that they are showing you on that particular graph. Because they're expressing their data as a log function, it doesn't seem like 0.31%. And that's the problem with visualizing. Unless you think in logs, you don't realize that 0.3% looks as big as it does on that particular scale. Their control final, again, illustrates that there's no real difference. But then when they add the E treatment or the E-scaffolding and they apply the current, what you see is the total biofilms drop down, and you are effectively dropping that approximately 4 logs in both the total biofilms and the total biofilms with the 6-hour treatment with the Cipro. But then on the population alone, with the E-scaffold on the persister cells, all those persister cells are gone. So it seems that the peroxide is a much more potent killing agent on persister cells than the vegetative cells, because they're only generating a very low concentration of hydrogen peroxide, not enough hydrogen peroxide that like your mother would pour on your cut to sanitize your wound when you skinned your knee, but it's just a low concentration of peroxide. And so their final result that they present in this particular paper is illustrating what the E-scaffold is actually doing and how it's permeating the membrane. And what they use here is some of these great membrane dyes that require an energized membrane in order to be excluded. And what they are measuring is the presence of fluorescence. And the way they did that is they took untreated cells and they added the dye to it. And the dyes that they are using are, in the case of the fluorescence, they're using 3,3' dipropyl thiocarbocyanide iodide, and they are measuring this. And what you see is that the E-scaffold treated cells accumulate much more fluorescence, which is suggestive that the membrane potential is gone and the cells are made wholly, and the dye is diffusing into the cell. And then they have as their control cells that have not been exposed to the E-scaffolding but were treated with a low concentration of exogenous hydrogen peroxide, and the data are indeed very similar. And so again, they then show us a delightful set of electronic micrographs that shows us how the E-scaffold treated cells are really being actively damaged by this in situ peroxide that's being generated. And similarly, when you compare it to the exogenously hydrogen peroxide treated biofilm cells, you again see similar damage in this system. So in sum, what these authors have been able to show us is that you can add a small amount of electric current to a biofilm-based infection, and you can reduce the concentration of antibiotic that's needed to clear the biofilm-based infection. So as Elio often says, stay tuned for new treatment modalities. The carbon fiber dressings that these authors were using are already being used for topical applications for wound dressings, and it opens up this whole new avenue for being able to treat these chronic and persistent biofilm-based infections. This is especially important when you look at individuals who develop these chronic biofilm infections of Pseudomonas, and the population that's at greatest risk are, of course, the kids who develop cystic fibrosis and have these chronic Pseudomonas-based biofilm infections.
Michael, how would you use this in a clinical setting with the current?
Well, since this is the Consumer Electronics Show, and some of the new stuff that's coming out is wireless charging for your phone, where your phone doesn't even have to be connected to a wire.
Mm-hmm.
I can well imagine that if we can make some of these particles that you could attach an antibody to, that could conduct a current where you could effectively have them go after some of this EPS where it could attach. You could add those particles to the bloodstream. They would seek out the biofilm with the antibody, and then you would just apply some of this wireless charging current. And it doesn't have to be very much, and you could then lower the concentration of antibiotic required.
I can imagine them wheeling in a little portable charger into your room in the hospital, and the whole
swallow
room
this.
You don't even have to swallow it. You just sit in the room, and the same way that your phone charges, like Michael says, in a wireless situation. Yeah, that's very cool.
We're already doing that with the iWatch to detect your heart rate and these other things. And, we're of the generation that wasn't as electronically trained as the generation coming into our classrooms today. And so the sky is the limit, which is why I wanted to bring it as a snippet rather than the full-blown paper, because I really want to stimulate interest to figure out how we can bring the electronic world to treating infections. When I first suggested this to Elio, I ran it by him. He was the cautionary tale. He said, more likely for a snippet than not. But
well, let's talk about specificity, because one of the reasons antibiotics are such miracle drugs is that they target pathways that we don't have, that mammalian cells don't have.
Correct.
But hydrogen peroxide doesn't care what protein or DNA or lipid it's attacking. So specificity and really targeting this constant source of hydrogen peroxide is going to be key.
And I think that's why I suggested using an antibody to the EPS or something along those lines that would somehow find the infection.
And then you'd think that the hydrogen peroxide would just react with whatever it comes in contact with first. It wouldn't diffuse away and attack, red blood cells or whatever.
I think it's limited by the current of how much current you can generate in the local environment. And that's why they've been playing with their potentiostat. I looked at some of their earlier work where they were playing with the potentiostat, and I think this is the time where we need to bring in an electrical engineer to help us begin to think about these things.
Of course. All right. Thank you, Michael.
Thank you, Vincent.
The first
Michele.
First TWiM snippet of 2017. That's a historic moment.
It is.
This episode is brought to you by CuriosityStream, the world's first ad-free nonfiction streaming service. They have over 1,500 titles and 600 hours of content. It was founded by John Hendricks, who used to be with the Discovery Channel, and that means you're going to get access to real science shows. So they have a variety of documentaries of different sorts, which you could watch on their website, curiositystream.com, or in using any of those devices that interface the internet with your TV, like the Apple TV or the Roku device. It's available all over the world, and they have a wide variety of science and technology, nature history, a lot of other topics. They also have interviews and lectures. Some examples of what they have: Stephen Hawking's favorite places. This is a new documentary in which he pilots a CGI spaceship across the universe and stops at his favorite destinations. Another series called Digits, a three-parter hosted by Derek Mueller, who created the YouTube science channel Veritasium. It explores online safety and security. Deep Time History is a three-part documentary on the universe's 14 billion year history. Underwater Wonders of the National Parks. It's a 7-part series that takes you underneath the bodies of waters in those parks. They also have super high definition, so-called 4K material. They have about 50 hours of 4K content as well. They offer monthly and annual plans starting at just $2.99 a month, less than a cup of coffee, or a single title on some of the competing platforms. Check out curiositystream.com/microbe and use the promo code microbe during sign-up to get unlimited access to the world's top documentaries and nonfiction series completely free for the first 60 days. That's 2 entire months free of one of the largest nonfiction 4K libraries around. Just go to curiositystream.com/microbe and use the offer code microbe at sign-up. We thank CuriosityStream for their support of TWiM. They've supported us for the past 6 months, for which we're very grateful. And you won't hear them for a while. I suppose our ad contract is over, but we really appreciate their support. All right, I want to tell you about a very interesting paper published in Cell. Now, we often don't do viruses here on TWiM, but now and then we do, especially if They involve viruses that infect bacteria, so-called bacteriophages. And that's what this paper is about. It's called Acquisition of Phage Sensitivity by Bacteria Through Exchange of Phage Receptors. And the authors are Zipilovich, Habusha, and Ben Yehuda from the Hebrew University of Jerusalem in Israel. And, as everyone knows, phages are viruses that infect bacteria. They are possibly the most abundant and diverse entities on the planet. Huge numbers, 10 to the 30th in just the oceans. And they have a big impact on shaping bacterial communities. They kill some bacteria, of course. That's called lytic replication and not others. They can also mediate horizontal gene transfer, big-time transfer of DNA via phages. Of course, sometimes when a phage infects a cell, it becomes lysogenic. It integrates into host DNA. And then when it excises, it can take a piece of host DNA with it and bring it into the next cell. That's called transduction. They do that a lot, and they bring DNA from cell to cell. Now, like most viruses, infection of bacteria with phages begins when the virus is attached to a cell receptor, and this initial attachment and entry step confers specificity to infection. Most phages are host-specific. They only infect a limited set of strains within a species. There's some exceptions, of course, but the specificity is determined at the level of attachment to a cell receptor. Now, this paper deals with phages that infect the gram-positive soil bacterium Bacillus subtilis. And for me, this brings me back, 35 years, because my wife did her PhD thesis on a phage of Bacillus subtilis.
Oh, cool.
Her phage was Phe105. And the phages in this paper, the main one is called SPP1. This is a double-stranded DNA phage of Bacillus subtilis, and it's composed of an icosahedral head where the DNA is kept in a very long, non-contractile tail, and at the end of the tail is a spike, and that's how the phage attaches to the host cell. There's some other phages that are mentioned in this paper and used as controls. Another one is Phe29, another subtilis phage that's well-studied. Similar, has an icosahedral head, but a very, very short tail, and some others as well. Now, SPP1 attaches to host cell. The tail tip has proteins in it that first attach to polyglycosylated teichoic acids, GTA. And these are anchored in the bacterial membrane by glycolipid or covalently attached to peptidoglycan. So there's this first attachment step, which is reversible. And then after that, the phage then binds to a membrane protein called YUEB, and that's irreversible and leads to injection of the DNA into the cell. Okay, so there are two components that are important for entry, GTA and UB, and these are going to play roles here. Now, what they do in this paper is ask, if you have mixed populations of cells that are susceptible to infection, so susceptible in virology parlance means the cell has a receptor for the virus and it can get in. That's susceptibility. Permissivity is what happens beyond the entry step. So I'm very specific about that. Some people are loose about the way they use those words, but having written a textbook, it's important. It's important to use the same word the same way throughout, right, Michele?
Oh, yeah.
Because if you mix it up, people go crazy. So susceptibility determines whether you have a resistant or a susceptible cell. And they mixed them together in this paper because that's the way they occur in nature, of course. We want to see what happens. So they have resistant cells. These are all Bacillus subtilis cells. And the resistant ones are simply lacking the UB gene, YUEB. That's the other membrane protein needed for infection. And they have made these resistant cells produce GFP, green fluorescent protein. So you could look at them in a microscope and see if they're there or not, see if they're healthy or if they're lysing. So if you take our cells alone and you infect them with SPP1, nothing happens because they don't have receptors. And they have another variant, the S cells, which are susceptible, and they have the UB gene. They're able to be infected. Of course, they're killed by infection. Interestingly, if you mix S and R cells and then infect with the virus, the R cells clearly go down after infection. And it's dependent on the presence of S cells, right? If you take out the S cells, the R cells do not go away upon infection.
To me, that was the most striking like that's what hooked me on this paper. When
Yeah.
they found they had to have the S cells in there.
Right.
So it wasn't just a random rare variant
Exactly.
of R cells.
Right. It could be a variant
infected.
that's acquiring susceptibility, but it's clearly dependent on another bacterial host. And they also do the same experiment with Phe29 and SPO1, which are two other Bacillus subtilis phages that use the same receptors, same effect. They will infect resistant cells in the presence of S cells. The next set of experiments are kind of interesting. They can make susceptible cells without DNA. They can take the DNA out of them. And of course, if you try and infect these with a phage won't replicate because there's no DNA and there's no metabolism, protein synthesis going on in there, so the phage won't replicate. But they can be lysed from outside. So if you had phages lyse hosts by making lysis proteins, and you can produce those and purify them. If you added those to these cells without DNA, the cells would lyse. So they can't be infected, but they're good ways to track the production of lytic proteins. So what they do is they make a phage encoding one of the lysins. This is the protein that lyses the cell, fused to yellow fluorescent protein, because they want to visualize the release of the lysin from infected cells. And when they infect susceptible cells with this particular phage, they can see the protein is produced. Upon lysis of the cells, it is released from the cells and combined to the surface of neighboring uninfected cells. And if these are resistant cells, it can make them lyse. And they can actually see that these green resistant cells are bound on the surface by this yellow lysin, and they're getting lysed. So the suggestion is that perhaps something's coming out of the infected cells and lysing the other ones. They also made cells with an inducible yellow lysin gene in them. So these are Bacillus subtilis which don't have the lysin gene normally, right? It's a phage gene. They put one in that's linked to a yellow fluorescent protein, induce the synthesis of this gene. They treat the cells with chloroform to release the lysin. Protein gets out, it binds to neighboring cells and lyses them. And remember, there's no phage in this experiment. It's just lysin being produced and released from the bacteria. That's enough to lyse neighboring cells. And both R and S cells can be lysed this way, of course, because there's no phage involved. All right, so where is this going? When they mix the lysin producer cells with 3 different Bacillus species and also Staph aureus, and these are all insensitive normally to SPP1 phage, they're all lysed. So lysis from without, which is what this is called, can impact communities composed of different bacteria, irrespective of their phage sensitivity. It's all about the released lysins. So that's one idea that they have here, is that lysins released from infected cells in the population might be from the S cells, might be lysing the R cells. Now, they noticed that in some of the infections of mixed S and R cultures, S susceptible, R resistant, with their yellow lysin phages, sometimes the R cells actually got infected, right? So you can tell this because these cells are carrying the lysin RFP gene, whereas it shouldn't be in there. The lysin RFP was engineered into the phage, so if that is being produced in the R cells, they have to be getting infected. They never see this in pure cultures of resistant bacteria.
And the paper includes panels, really beautiful fluorescence microscopy panels, so that you can follow these events in the population.
Very pretty pictures, yeah. Very visual. Now, this effect of getting yellow lysin in the R cells is more pronounced later in infection when most of the S cells are lysed. So that's a hint about what might be going on there. We'll get back to that later. All right, moving on. Now they look at another way of assaying phage infection, and that is by the production of replication foci. What they do is they take the gene encoding the major tail protein of the phage. This would be something that's needed for assembly of the phages. They link it up to green fluorescent protein, and they make a cell producing this protein, and the protein is diffusely produced in the bacteria. But when they infect these bacteria with phage, later in infection, the protein distribution changes from diffuse to discrete foci in the cytoplasm, and they think these are assembly sites. So, the phage is making its own assembly protein, but it will also utilize the green labeled tail protein produced by the cell, and those coalesce into assembly foci.
And those images are quite striking. There's no question about that result.
Now, the cool part is when they take a mixed culture of S and R cells, and the cells are all producing this green tail protein, they see the same foci form in R cells in a mixed culture. And it's more evidence that the R cells are being infected, right? Not just being lysed by reduced lysins, but because this tail protein is redistributing, they take that as evidence of infection. And they also provide a lot of other evidence of infection. They look for phage DNA in R cells by fluorescent in situ hybridization, and it's clearly there. So the DNA, the phage seems to be getting into R cells and replicating. One of the issues here is that you could ask, well, maybe some mutants of the phage are arising that can infect R cells. I think this is probably unlikely because
there's too many.
Yeah, there are too many infections, and it only happens in mixed cultures. If you take the S cells away, it doesn't happen. But they address it anyway. They take phage lysates and they pass them 5 times in mixed cultures, and then they take the 5th passage. It still cannot infect R cells alone. Just you need those S cells. They also show that other phages of Subtilis exhibit the same phenomenon. They can infect R cells in a mixed culture. They call this acquisition of sensitivity, and that's what's in the title of the paper, of course. They show that this effect can mediate transduction. They allow the phage to pick up an antibiotic-resistant gene in a susceptible cell, and they show that in an infected mixed RS culture, the gene can get into R cells. Very easy to assay antibiotic resistance, and they can show that the R cells pick it up. So it's clear that...
albeit at very low frequencies.
Extremely low frequencies, right? So we're now convinced that R cells can be infected in a mixed culture. How does it happen? So they're thinking maybe it's acquisition of the receptor from the sensitive cells. So to address this, they have a strain of bacillus producing yellow UB protein. Remember, that's the second protein you need for the phage to enter. And they use a technique called Fluorescence Recovery after Photobleaching. So the idea here is you have a sample which is fluorescent. You bleach part of it to eliminate the fluorescence. You shut off protein synthesis with an antibiotic, and then you see if the fluorescence comes back. If you can do this in a single cell, and that would happen when because the membrane is fluid, the protein can move in from other parts of the cell. But in this case, they're looking at neighboring cells and seeing if the protein can go from a fluorescent cell to a bleached cell. Kind of cool. So what they find is, in fact, recovery of irradiated cells within a few minutes, showing that receptor molecules are provided by nearby cells. When there are no non-irradiated cells nearby, they don't get any recovery. So they think that receptor proteins are being exchanged among neighbors.
And it requires proximity, right? Yeah.
It has to be close, and it requires protein synthesis, right? Mm-hmm. They also use a lectin called concanavalin A. This specifically binds GTA, which is the first binding component of the phage in the bacterial membrane, and they can show that con A binding is actually transferred to neighbor cells after bleaching. So not only is fluorescent UB transferred, but that GTA as well, the phage attachment molecule is provided. And finally, how are the proteins being exchanged among the bacteria? So they know, it's known in general that bacteria, uninfected bacteria, shed membrane vesicles, and these can fuse with other bacteria or even eukaryotic cells, and they're known to contain proteins, metabolites, even DNA. And they look in their cultures, and in fact, they find yellow UB on membrane vesicles from growing cells, and the phage can actually attach to them. And these membrane vesicles from S cells, when added to R cells, can confer phage susceptibility. But if you make membrane vesicles from R cells, it doesn't work. And in addition, the production of membrane vesicles is boosted when phages lyse the cells. So that makes sense because as the cells lyse, they're producing more membrane vesicles and they have the receptor protein in them. Finally, if you treat the membrane vesicles with chloroform, it blocks the ability of them to confer infectability to R cells. So that is what's going on. Looks like the membrane vesicles are transferring the receptor protein from the S to the R cells. And the final experiment is that they take R cells that lack both the UB gene. So far the R cells that we've been talking about lack only UB. They also delete the genes for GTAB. in a mixed culture, these can still be infected. So the membrane vesicles are apparently transferring both GTA and UB from the S to the resistant cells. And this can happen, this kind of transfer can happen to cells that are not normally infected, like Bacillus cereus and others. They find that just co-culturing or adding MVAs to these cells will allow the phage to attach. Now, in these cases, they don't look for replication because beyond entry, you have to do other things to be able to a phage to replicate in a different host. There are restriction systems that prevent phages that grow in one bacteria to infect another. But, the attachment and entry step, which is a major block, clearly can be overcome simply by having the protein transferred in these vesicles.
And it's also a race, too. It's also a race, too, because it's how fast the restriction endonucleases are. Because once the phage begins to replicate its DNA, it can then tag it with the appropriate. So it's a race. Some phage probably win out. So it's an evolutionary advantage to that phage to do the swap.
That's absolutely right. Yeah.
So the idea is these membrane vesicles that are shed from the infected cell or the lysed susceptible cell then intercalate into the membrane of neighbors.
Yeah, they fuse with the membrane of the neighboring cells, right?
Mm-hmm.
And that's how they deliver the protein.
Well, aren't we glad flu doesn't do this? Can you imagine if the flu virus was able to do this?
Well, that's a good question. I'm wondering if, and in fact, after I finished reading this paper, I'm thinking of experiments to test this for mammalian viruses. It's very easy. It's very easy to do, and I wouldn't be surprised because mammalian cells certainly make membrane vesicles.
Oh, yeah.
Right?
They're much
Exosomes,
better at it.
And they could have receptors, so you could temporarily confer susceptibility to a different cell. Yeah.
Yeah.
Now, one of the things they mention in the discussion, so they actually cite a paper of ours from the 1990s.
All right.
A paper of ours being cited in a phage paper. Oh, my gosh. What's happening to the world here in 2017? So they say what we did is we changed the tropism of polio by putting the receptor gene into transgenic mice. So transgenic mice cannot be infected with poliovirus because they don't have a receptor. But if you put the receptor gene in as a transgene, you can then infect the mice. So they're wondering if proteins, the exchange of proteins, not even genes, but the exchange of receptor proteins could have a similar effect. And I would say, yes, in a mouse, for example, if you put the polio receptor into any cell, you can get it infected. So if you put the protein or the gene in, I would guess that you would have the same effect. So really, this has lots of implications, of course, for bacterial infections, but I think it's a really interesting question whether this happens in mammalian cells as well.
You're going to have to go back and look at your cell paper from 1990.
It's so old. I'm so surprised they actually found it and cited it because, people have kind of a blindness to the past. They just go forward, which is the way science works. But it's really cool to see that, especially in a phage paper, right?
Yeah.
Now, one thing they say here is, so this transfer of the receptor can result in the transfer of antibiotic resistance genes, virulence genes into cells where they normally wouldn't get into, right, if they acquire the receptor in this way. And they say this should be considered when using phages as therapy, right? If you want to treat infections with phages, you have to be careful. Maybe you're going to introduce by transduction genes into cells that would not normally receive them. So it's a really interesting
And this high
idea.
degree of specificity that we talked about for the snippet might not be quite as specific.
Yeah.
If there can be promiscuous sharing of receptors in the population.
Yeah, it's a really provocative paper. I think it's quite interesting. It's new, and it has implications not just for bacteria and phages, but I think this has to be looked at for eukaryotic viruses, archaeal and eukaryotic viruses as well.
Yeah.
And would it provide an opportunity for the virus variants to be able to infect and actually establish a productive infection in new cells?
So once you get into a cell that you normally wouldn't be able to, maybe that gives you a foothold.
Some rare variant might be able to make a start.
Yep, absolutely. Absolutely. So I enjoyed that one. It was good.
Yeah, I liked that they were highlighting membrane vesicles because the bacteriologists, I think, don't appreciate the impact they can have on biology. So many membrane vesicles, as you point out, carry adhesins, some carry toxins, and they confuse not only with bacteria but also with host cells. So we can think of it as kind of an ancient secretion mechanism where microbes can deliver virulence factors directly into host cells, mammalian cells.
Right.
So I thought that was
absolutely
cool.
spooky. That's even worse than before.
Yeah, the barriers.
But it's happening. It's happening.
Oh, I know.
There are no barriers. Things can go back and forth. Really cool.
I like that they describe this in a nutshell as a transient and non-genetic process of horizontal gene transfer.
Yeah, when you think about it, it's quite interesting because the protein gets in, it's transient. That would be there transiently. But then if it lets phage in, then those genes can integrate.
Right.
The phage becomes lysogenic.
Yeah. Very interesting.
Well, that's their bottom line up front.
Yeah.
That's their bottom line up front.
So I was able to exchange emails with the first author. Elaine, help me out. Elaine?
Yeah, I looked it up how to pronounce it, actually. Let me, Elhanan.
Elhanan, and his last name?
Zipilovich.
So he is a graduate student at the University of Jerusalem, Hebrew University in Jerusalem, and he first got very excited about science when he took an introductory course in cell biology. And I think that really comes through here because although the paper is about phage, it's really about the cell biology of their host cells, which are bacteria. So he said one of the exciting moments in this research project was when he did the electron microscopy and found and could visualize the phage binding directly to the purified membrane vesicles. So that kind of allowed them to connect some dots and realize that they had a mechanism. He really enjoys science and especially appreciates that it is a field where your progress is dictated not by your class or your status, but you can achieve, and your accomplishments are really determined by hard work and persistence. So anyone, it's a meritocracy, in other words, anyone can come in, and if they work hard and are dedicated to a particular problem, they can advance. And his advice to junior scientists is always to keep reading the literature and stay up to date, because that's where you can get new ideas for biological pathways or also techniques to bring to bear. So he is married to a woman, and they have three kids, 2 boys and a girl. So I'm sure he's got a very full and satisfying life in and out of the lab.
Wow, I'm impressed.
Yeah.
And he's a graduate student.
I believe he is, yes.
Wow. Well, the good thing is he will be young when his kids are older.
That's right.
As opposed to me, who's old and my kids are still young.
Yeah. I had both my children as a PhD student, and it's definitely doable.
Oh, yes. In fact, Marion Carlson did the same.
Yep.
So
she was my advisor.
Yep.
Maybe
she influenced you in other ways. Who knows?
Could be.
Well, thank you for that exchange.
Yeah.
We have 2 emails. One is from Peter, who writes, "Greetings, TWiM team. Thinking about the revised estimates for the number of bacterial and human cells in the body from 10 to 1 to about 1 to 1, which, by the way, we covered in a TWiM some time ago. I was wondering what the microbiome cell-to-self ratio is for different species. I presume that it would depend on diet, with herbivores having a higher proportion of microbial cells than omnivores, and that omnivores would have a higher proportion of microbial cells than carnivores. I would guess that the smallest gut microbiome would belong to hummingbirds that exist on nectar and insects. Has the size and diversity of the microbiomes of different species ever been investigated?"
This is a wonderful question for which I was scratching my head thinking about hummingbirds and nectar, because, they're effectively living off of a very simple molecule.
Yeah.
And yet at the same time, they still have to make a bird.
Mm-hmm.
And I just don't know if anyone's looked into that. So as Elio would say, stay tuned.
Yeah.
I think there is data for humans in that the diversity of your gut microbiome is influenced by diet.
Sure.
So if you're on a simple carbohydrate, high fat diet, your microbiome community is less diverse than if you eat a lot of plants and grains and fruits.
Right.
But I don't know if they have compared cross species, this ratio of the microbiome to self. I think that's a tough question. I don't know that we have that information.
I think you're right.
Even the one person who you might pester is ask Tom Schmidt, Michele, to see if he's asked the question about, because he's always looking at leopards, right?
Not that I know of.
Leopards? You mean
he was looking big cats.
Big cats,
He was looking at the microbiome of big cats, and that's, if anything, a carnivore. So he may have some insight into this.
I looked and I couldn't find anything. So I think you're right, Michele. It just hasn't been done. But, some of these insects, they have few cells, Like, let's take C. elegans with, 600 or 700 cells, right? They have far more bacteria than cells, probably.
Well, they eat bacteria. So,
well, you have to. That's not your microbiome, really, right?
No,
what you consume.
Well,
the ones that are resident that stay in you, but that could be transient as well. Who knows? But I can imagine situations where the ratio is reversed, where there are lots more bacteria than. Anyway, I don't know. I didn't find anything, but there must be listeners who know more. So weigh in. We'd love to hear from you. And Roman writes, "Dear TWiM hosts, love your podcast. Keep it up. Could you give me a name or link to the soundtrack you use? You say it's by Ronald Jenkins, but it's still hard to find." Well, Roman, if you go to his website and listen to every song, you would find it, which is what I did because I couldn't remember the names. It wasn't so hard. You can play all of his songs on his website. The introduction is called Remix to a Remix. It's from his first CD, which is called Ronald Jenkins. And the outro, the ending music, is called Stay Crunchy, and that is from his CD called Disorganized Fun. And you can find all of that at ronaldjenkins.com. And it's all with permission, by the way. I asked Ronald Jenkins if we could use his music, and he loves it. He doesn't charge us anything. Good guy.
How did you find the music?
My older son, loves it. He's loved it for years. And when I was looking for it, I said to him, "You think this would be good?" And we sat down and listened to a bunch of tracks, and use it on TWiM and on TWiP as well. I used 2 different tracks from Ronald Jenkins. I like them. I think they're good.
What do you use for TWiV?
For TWiV, that was a long time ago, and I just used 2 free tracks that I found in GarageBand, which was an Apple program.
Okay.
And at some point, I'd like to change them because they're a little old. But now, all the new podcasts I make, they're websites where you can actually buy licenses to music and use them, and you can get a lot of cool stuff. That's what I do now. All right, that is TWiM 143. You can find it at iTunes. You can find it at, asm.org. Those of you who go to microbeworld.org, that's eventually going to go away, and all the podcasts will live at asm.org. Or if you listen on your cell phone, of course, you get a podcast app and you just subscribe to the podcast for free. Think about becoming a patron of TWiM and all the other Microbe TV family of science shows. Go to microbe.tv/contribute. We have several ways that you can help us out. And please do send your questions and comments to twim@microbe.tv. Now, I have a book to give away to our listeners.
Oh, that's right. We forgot about that, Michele.
Well, I left it all the way to the end, so
I know.
People would listen or skip through. I don't know. All right, I have a brand new copy of Manual of Clinical Microbiology.
Ooh, that's a good one.
This is the 11th edition. It just came out. It's published by ASM Press. It's in 2 volumes. I don't know, each volume weighs about 5 pounds. It's really an impressive book for people who do clinical microbiology. It's got chapters on every isolation technique through all the bacteria, antibacterial agents, and much, much more. Many, many authors, of course, have contributed to this. And let me just read for you. The Manual of Clinical Microbiology is the most authoritative reference text in the field of clinical microbiology. So if you would like a copy of this, it's brand new. I'm happy to send it to you. All you have to do is send an email with the subject line, "I love microbes," and the 19th person to send me that email will win a copy of this book.
And that's at twim@microbe.tv?
That's right.
It's the email address? Okay.
That's correct. And I'll do this regularly because, as I said, I get a lot of books from ASM Press. I'd love to share them with our listeners. I think they would love to have them. So don't forget, try that out. Now, it's tough to figure out when the 19th will come in. Over on TWiM, we have a running contest now for the third week. And that one was the 23rd email. We still haven't reached it. So you have to just try. Michele Swanson is at the University of Michigan. Thank you, Michele.
Thank you.
Michael Schmidt is at the Medical University of South Carolina. Thank you, Michael.
Thanks, Vincent and Michele.
And I'm Vincent Racaniello. You can find me at virology.ws. I'd like to thank the American Society for Microbiology for their support of TWiM, and Chris Condian and Ray Ortega for their technical help. I also want to thank the sponsor of this episode, CuriosityStream. The music you hear on TWiM is composed and performed by Ronald Jenkins. You can find his work at ronaldjenkins.com. Thanks for listening, everyone. We'll see you next time on This Week in Microbiology.